Hi everyone, myself Unnimaya from First MSc Biology. Today we are discussing about the topic Immuno-fluorescence Microscopy. Introduction. Immunofluorescence is a powerful technique that utilizes fluorescent-labeled antibodies to dictate a specific target antigens. The fluorescent molecule absorbs the light of one wavelength and emits the light of another wavelength. If antibody molecules are tagged with a fluorescent dye or fluorophore, the immune complex containing these fluorescent-labeled antibodies can be detected by the colored light emission when excited by the light of appropriate wavelength. The antibody molecules bound to antigen in cells or tissue section can similarly be visualized. This technique is known as immunofluorescence. The most commonly used fluorescent dyes are fluorescein and rhodamide. Other dyes such as phycoerythrin and phycobilivprotein are also used nowadays. Immunofluorescence are of two types, direct and indirect immunofluorescence. Direct fluorescent antibody method, the specific antibody is directly conjugated with fluorophore. Indirect fluorescent antibody method, the primary antibody is unlabeled and is detected with a fluorophore labeled secondary antibody. It is a two-step process in which primary unlabeled antibody binds to the target, after which a fluorophore labeled secondary antibody is used to detect the primary antibody. Diagrammatic representation of direct and indirect immunofluorescence. Direct immunofluorescence, it is very simple and common procedure. Antigen is fixed on the slide. The fluorescent labeled antibody is layered over it. Slide is washed to remove unattached antibodies. Examined under UV light in a fluorescent microscope. The site where the antibody attached to the antigen will shows apple green fluorescence. Advantages of direct immunofluorescence. This technique is several advantages over indirect immunofluorescence because the direct conjugation of antibody to the fluorophore. This reduces the number of steps in the staining procedure, making the process faster and can reduce the background signals by avoiding some issues with cross-reactivity. Uses. Used to detect viral parasitic tumor antigens from patient specimens or monolayer of the cell. Indirect immunofluorescence. Indirect test is a double-layer technique. The unlabeled antibodies applied directly to the substrate. Treated with a fluorophore conjugated with anti-immunoglobulin serum. Advantages of indirect immunofluorescence. Give an amplification effect. Requires only one labeled antibody to identify many protein. Uses. Detect autoantibodies such as anti-nuclear antibodies in autoimmune diseases. Identify the class of given antibody by using fluorescent antibody specific for different immunoglobulin isotopes. Thank you.